It's like 1%. Yeah, that's good. That could be it.
You cannot get it. It's fine. Sometimes, yes, sometimes no. Exactly.
That was was it? What did you say? Okay, that makes sense. Well, some of them are repeats, so I don't know. But you are correct twice. I'm sure you hear but they're all different. Oh, then I should read them all. But I just kind of read them. Yeah, different. Challenging. And I was like,
I can cross this room.
I'm betting because you usually go straight to like if you were at work, you should have been so you want to like be caught on the because okay, so they were here when they saw it. Tim's doing out. Tim's she didn't understand mommy. It's like so much for me. That's not true. With the title, it's gold. I don't know. I like dynamite.
Okay, hi everybody. Welcome to the 2017 Kendall Lecture in honor of Dr. John Kendall, an esteemed faculty member in the Department of Psychological Science and former president of the college. My name is Patricia Reeder. I'm an assistant professor in the department, and it's my pleasure today to introduce this year's Kendall speaker, Dr. Jed Ellison. Dr. Ellison is an assistant professor at the Institute of Child Development at the University of Minnesota. He did his undergraduate work at the University of Utah and earned degrees in psychology and English literature. Completed his graduate work in psychology at the University of North Carolina, Chapel Hill. And after a postdoc at the California Institute of Technology, he decided to move to the frozen tundra of Minnesota and had his current position at the University of Minnesota. Dr. Ellison's research focuses on identifying neural and behavioral markers that could help indicate young infants who are at risk for autism spectrum disorders. Through behavioral assessments, eye tracking interviews, magnetic resonance imaging, Dr. Ellison is enabling a better understanding of brain and behavior development. So this research has the potential to reduce the age of first diagnosis of autism, thus allowing earlier interventions for infants at risk of the disorder. So for this work, Dr. Ellison has received a number of grants and awards, including a Biobehavioral Research Award for Innovative New Scientist, the BRAINS Award, from the National Institute of Mental Health. He was named an APS Rising Star, and he received the Early Career Research Contribution Award from the Society for Research in Child Development. So please join me in welcoming Dr. Jed Ellison for his talk, Early Brain and Behavioral Development in Autism for its Pre-Symptomatic Prediction.
Thank you. It's a pleasure to be here. I'm going to present some data and some materials today that interestingly enough, had its humble beginnings when I was a junior or a senior as an undergrad, so it was great to participate in the ceremony earlier. But I've been thinking about these ideas for a long time, and they really kind of their initial seed was planted when I was an undergraduate at the University of Utah. I needed to complete my senior thesis, and I found my way to the Department of Psychiatry actually, because that's where the autism research was going on, and I carried out a study, a small study, and 30 kids who had brain imaging data collected as part of the study, another project, a well-funded project, and I just carved up the sample a little bit differently. I was examining the presence of attention deficit hyperactivity disorder in kids with autism, and then a group of kids with autism who didn't show these attentional deficits, and then a typically developed control group, and I examined brain structure differences between those groups. And I've been studying attention in autism ever since. Right around the time I finished my undergraduate degree, the first publication characterizing development in infant siblings of children with autism was published, and that really kind of solidified my interest in studying babies and studying infants at risk for developing autism. Thanks to Marie Walker and Heather Unger and Carrie Humble for helping me get situated and making this an easy trip thus far. So here's the outline for this evening. So I'll give you a couple of slides on what autism is, contemporary conceptualization of autism. I'll tell you a little bit about early identification methods. We'll walk through some findings on early identification. We'll talk briefly about pre-symptomatic prediction, what that might look like. We'll cover a little bit on what prevention of autism might look like. And then we'll give you some future tips. So what is autism? Everybody here, probably, I'm nearly certain that everybody here knows someone with autism. Public awareness at this point is quite good. And many of you will know that autism is a behaviorally defined disorder. There are two major behavioral constellations or behavioral constructs that define autism, and this includes impairments in social communication or interaction. And the second broad domain is restricted and repetitive behavior. So this is a change as of a few years ago with the publication of the new Diagnostic and Statistical Manual, the DSM-5. Beginning in 1994, you used to think about autism as this tripartite model of social impairments, communication deficits, and restricted and repetitive behavior. But in the DSM-5, they put the social communication, language impairment, and social communication deficits together, and just call it social communication deficits. But this doesn't really represent what autism is, right? These broad domains. In reality, autism is much more complex. And it's likely not a single phenomenon, but rather multiple different types of disorders that tend to have some convergence in this behavioral manifestation. So when we talk about phenotypic heterogeneity, it's just a jargony term for variability in how the disorders manifest. A couple of these bubbles that I want to highlight. So to highlight the variability and phenotypic heterogeneity. About 30 to 40% of individuals with autism have co-occurring intellectual disability. As the prevalence estimates have increased over years, everybody sees these reports. One didn't come out this April, but the past 5 years or every other year, the past 10 years, new reports come out from the CDC reporting an increase in prevalence. We'll talk briefly about that. The prevalence of individuals with autism and co-occurring intellectual disability has not changed essentially since 1989, since our first, kind of the earliest, best prevalence estimates. So what does that mean? That means that what we're doing is identifying more high-functioning individuals with autism. That also means that the old rates, we used to think that about 60 to 70% of individuals with autism had this co-occurring intellectual disability. Another bubble that I'll highlight is seizure disorder. This has remained pretty stable as well. About 30% of individuals with autism have co-occurring seizure disorder. This was particularly important in the history of autism research because it helped solidify autism as a brain disorder, the identification or characterization of this associated seizure disorder. So in the 60s, when people were talking about psychogenic causes or autism resulting from bad parenting, someone named Bernie Rimland, a neurologist at UCLA, actually wrote a book and collected the first series of data characterizing the association with seizure disorder, and essentially postulating that autism is this disorder of brain function and brain development. A couple of other bubbles that are hot, I'll highlight. Individuals with autism often show co-occurring behavioral comorbidities or deficits. ADHD is commonly observed. Anxiety disorders are commonly observed. And this can make treatment very messy, and also characterization very messy, very challenging. But again, I wanted just to highlight some of these bubbles and these themes to kind of hit home the fact that autism is a heterogeneous disorder. It manifests in very variable ways. And so to the extent that at this point trying to talk about identifying a cure for autism doesn't really make sense, because we have to find cures and we don't know how many. It could be 10, it could be 20. There are multiple causes of autism. So there are other, rather than thinking about the major goal of being to cure autism, now the big movement is this idea of precision medicine and actually trying to embrace the heterogeneity and target treatments to individuals or smaller subsets of people. So what else do we know about autism? Mentioned the prevalence just briefly. So the most recent report from the CDC last year suggested about 1 in 68 8-year-olds are diagnosed with autism. I include this term, an estimate. It's important to remember that this is an estimate, and when these are reported in the CDC reports, you need to remember that this is the lower bound of an estimate. So it's probably anywhere between 1 in 68 and 1 in 250, something like that. So average age of first diagnosis across the sites that contribute to this data, there are about 12 sites around the country that contribute. The average age of first diagnosis is 46 months. North Carolina is doing really well. They've had state support for autism work for a long time, and the average age of diagnosis of autistic disorder is 37 months. In Arkansas, it's 53 months. A report came out last year from Minnesota. The average age of diagnosis in Minnesota is 54 months. So we're not doing as well as we think we could be. The cost, lifetime costs of rearing a child with autism are about $1.5 to $3 million more than rearing a typically developing child. So not only does autism result in substantial psychological burden on families and individuals, but also substantial economic burden as well. We do know that early behavioral interventions do work, especially in intense interventions. I'll talk about one of those later in the talk. And I mentioned this idea, this kind of broader movement in it's a broader movement in psychiatry, but people in autism are really beginning to embrace this approach as well because of the vast heterogeneity that we see in autism. So this idea of precision medicine that was really born out of oncology and cancer research is really trying to use more biological information to personalize the treatment approach. So, and I'll have a slide on that specifically shortly. So here's a representation of an individual with autism. We all know people with autism. This comes from a series that Discovery Channel produced called Autism by 6, and it's about a family with 6 children with autism in Utah. Let me plug in my audio.
Bobby Bobby is 14 years old. He borders on high functioning autism and Asperger's syndrome, and he is from my first marriage.
He's very shy. He's a very shy boy. You might be termed as a typical wallflower type, where he just kind of like just fades back.
He works really hard. I mean, he knows how to do the most things to help us out, and he's really good at it.
And his big thing is detailing. He just will really good do exactly every little piece that he has to do. He'll follow instructions to the T. And so when he's cleaning, it's like he does every last little bit.
Bobby does shoulder a lot of burden in being, as the oldest child in this family.
It's like obvious to be done. Someone else did it. So I'm the one. Yeah, I'm taking the bar right now.
Bobby's focus is great. He will come home from school, jump on the computer, get on the internet, and he'll look and see how his grades were for that day.
Wow. Oh my goodness, thanks, Bob.
It's still me.
That's really good. Just an A minus? We talk about just an A minus. Oh, I had to work really hard just to get like a B average in high school. You were super smart, Bob.
Equations on the board, and it says in the first one, we're gonna solve using seven.
Bobby's been diagnosed with Asperger's syndrome, which by definition means that you're in the high functioning range. Most people with Asperger's syndrome or mild forms of autism are not good at picking up social cues.
Hi, come on in, have a seat.
What I'm trying to find out from Bobby is how he perceives social interactions, how well he can do a back and forth conversation.
Oh, all right. What else do you like to do for fun?
Watch TV.
Yeah? What kind of shows do you like?
SpongeBob.
Oh, I love SpongeBob. Do you have a favorite character in SpongeBob?
Squidward.
Really? That's cute. I have a favorite character in SpongeBob.
Am I supposed to guess?
Most people with autism don't initiate a lot of friendships. Have you ever had a best friend?
I still have best friends.
When I ask him about friendships, I'm trying to see what his concept of a friend is.
I have some best friends in 4th period.
Yeah?
And then there's one kid,
Uh-huh.
he's like the class clown.
Really?
Yeah.
Do you ever see him outside of school, like after school or on the weekends?
No, it's like only in school.
From what he tells me, these kids sound a little more like acquaintances than true friends. I wasn't surprised when he told me about his friends and then he said, when I asked him more, like, do they come over? I wasn't surprised when he said no. But I also wasn't surprised that he really considered them good friends. I get that a lot from kids with Asperger's syndrome. For Bobby, the amount of social contact he has with his friends right now is probably very satisfying to him, and that's a good thing. The trick is all of his social contact is structured by other people, and when he's an adult and he's no longer at school, he won't have those opportunities.
Okay, I like that clip for a couple of reasons. So one is you get to see the deficits in communication, communicative pragmatics, quite explicitly, right? So communicative pragmatics are the unwritten rules of communicating. How long do I pause to allow you to respond when I ask you a question? What are we communicating with eye contact and gesture? The other reason I like showing this is that this is, I like to think that we're working on kind of the cutting edge of early identification. The next frontier is really understanding the emergence to adulthood and what is happening with adults with autism and how they age when age differently than normative samples. There's so little data on adults that it's a little scary. The last thing that I want to mention is that individuals like Bobby, who have very high functioning, they have a lot of skills and very kind of subtle social deficits. These are not kids that we're able to identify very early. And in fact, this is another kind of blind spot in some of the research that we conduct is these children are identified much later and diagnosed much later. And so this is an area of challenge for us to, my colleagues and I, trying to figure out how to identify these individuals who will be diagnosed with autism but have high IQs and be doing pretty well, as early as 2 or 3 years of age. So what are some of the methods that we use for early identification? But before we get there, let's set the stage. So again, average age of diagnosis is 12 months or so. We generally make a reliable stable diagnosis around between 18 and 36 months of age. How are early signs detected? Oftentimes they're observed by parents, and so parents are concerned. Other times we have concerns from primary care providers, so pediatricians. The other two methods are what I'll focus on, some of the research that we do. So population-based screening, where the general approach is to ask parents when they come into a well-child checkup at 18 months or 24 months to fill out a 20-item parent-report questionnaire. So 20 items, 20 questions about, does your child do this, yes or no? And then tally those scores, and if you exceed a certain threshold, then you're invited to come back for a phase 2 screening. And then we also work in monitoring high risk, so I'll tell you more about that design. So just went over this, but the traditional approach to population-based screening, so the American Academy of Pediatrics recommended full surveillance and screening of 18 to 24-month-olds. This recommendation was made in 2007. Population-based screening and the instruments that we have, they work okay. They have pretty good sensitivity, so that means that they're identifying most of the kids who are developing atypically and many who go on to develop autism. But they're not specific, so they're not very cost-effective. You're identifying a lot of kids who don't have any problems, who are going to develop just fine. The other problem with this data, these data, and this is kind of a technical problem, but it's a huge problem, is that they only, the vast majority of studies have only followed individuals who screen positive. And so when you're dealing with a sample of 20,000 and say, 1,000 or 2,000 screen positive, and those are the individuals that you follow up, you make an assumption about those other 18,000, right? And we have no data on negative predictive value or what's happening with those other 18,000. How many of those are misclassified? So that's a huge problem with the research to date on population-based screening. So we are implementing a slightly new approach in the Twin Cities. I'm going to share some data with you that I haven't. You guys are the first to see it. So it's brand new data. It's not published. And it's still a little rough around the edges, but it will give you an idea of how we're thinking about early screening. So what we're doing instead of minimizing burden totally on parents and just asking them to complete 20 items while they're sitting in the foyer of the pediatrician's office, we're actually incentivizing parents to provide more information about their child's development. So what we're saying to parents, we're saying, look, we'll send you an e-gift card to Target if you complete 45 minutes worth of questionnaire data about your child's development, and we'll also enter your name into a drawing for a $50 gift card. And we're getting great response rates from families, community sample in the Twin Cities. But what this is going to allow us to do is use more information about their child's development to make a more precise prediction. And so, and then we use these fancy statistical techniques to derive what we think of, what we're calling risk profiles. So for those of you who are statistically inclined, we're using a factor mixture model. So essentially we've got 3 questionnaires. 2 of them are brand new, and very innovative. One is characterizing patterns of repetitive behavior in toddlers, okay, 18-month-olds and 18 to 50-month-olds, but we're starting with 18-month-olds. So we have one questionnaire on repetitive behaviors and all of the subscales of repetitive behaviors. So repetitive motor movements, self-injurious behaviors, rituals and routines, kind of the obsessive type behaviors or rigid adherence to routine type behaviors. In young kids, we call them just right behaviors, where kids need to have a routine at bedtime or around brushing teeth. It's very common in developing kids. And then the fourth subcomponent of the repetitive behavior scale is interest. It's around play behavior and whether play is restricted in certain ways. And then we have a questionnaire that's dedicated to reciprocal social behavior. And then we have a questionnaire about language development. That's kind of a workhorse measure called the McCarthy-Bates Communicative Development Inventories. So we use all of this information and essentially do a simultaneous factor analysis and linked class analysis. So we generate these dimensional factors that then that capture common variance in these questionnaires. And then we use this information to derive latent classes. So these are data-driven profiles of kids, okay? Classes of kids who show common profiles on these questionnaires. And we verify that, in a sample, we verified in a sample of 1,570, that this 5-class solution does capture, or represent kind of the variability in behavior pretty well. And what you see is, if you look in this 5 classes, a couple ways to verify that the number of classes that you're getting right is you look at whether it's 3 classes or 4 classes, 5 classes, 6 classes, and whether some of the kids track together. And so you'll see in this 5-class solution, we've got a couple of very small classes of kids who seem to be showing very different patterns or profiles of behavior. And then we've got a class of about 80, which is about 4% of the sample. So about 6% of the sample is captured by class 1, class 2, and class 4. So the question is, okay, that's cool, but is it meaningful, right? So we can look back at our data and look for stratification that would indicate risk. So for example, in our repetitive behavior measurements, on these 5 bars, you're showing, this is the one I like to highlight because you're getting great stratification. Our 3 smallest groups are showing the most kind of restricted interest or restricted play behavior between 18 and 24 months. Our 3 risk groups are also showing the most kind of rigidity or adherence to routines, or obsessive type behaviors. Interestingly, we're also getting some a bit more self-directed or self-injurious behaviors in these kids as well. So think about self-injurious behaviors in a toddler. You think about a kid who it happens quite often. I have a 22-month-old at home. When a tantrum's being thrown, if there's any head banging on the floor or something like that. And then there's much more variability in repetitive motor behaviors, but we still see slightly higher scores in these 2 risk groups. So another way, this was the data internal to generate the classes. But we can also look at data external. So we also followed up about 120 of these kids and asked parents to complete another questionnaire, a separate questionnaire, about early emerging internalizing symptomatology, externalizing symptomatology, this construct called dysfunction that captures some autistic-like behavior. And again, we're seeing stratification in our groups. So we think that this data-driven approach to characterizing risk profiles early in development may allow more precise clinical recommendations for these kids. So just what is precision medicine, just so everybody can take that keyword home. Many of you will know what it is. But essentially, it's very simple. It's the right intervention at the right dose for the right person at the right time. As applied to screening, the way that we're thinking about it is essentially the right clinical recommendation for the right child at the right time. Okay? So another approach to early identification is monitoring infants who are at high risk. So about 10 to 20% of younger siblings of children with autism will develop the disorder themselves. That number goes to about 30% if you have 2 older siblings with autism. As I mentioned at the outset, it wasn't too long ago that the first paper was published on studying these infant siblings with children with autism. It was really a game changer for researchers because it afforded the opportunity to track autism from its very earliest manifestation. So what are some findings? What do we know about early identification? Well, one take-home finding is that there are 0 overt behavioral markers of autism at 6 months of age. We've looked and looked and looked and looked, and there's nothing that will differentiate a 6-month-old who goes on to develop autism from one who does not. Now, what I'm citing here are several papers that have used sophisticated eye tracking technology. So you can identify some covert markers, and I'll give you one example in just a minute. But we have identified a number of behavioral risk markers at 12 months, including API decreased or reduced social interest, reduced shared positive affect, decreased or reduced social smiling. I bolded this one inconsistent or lack of responding to one's name because I haven't changed my story, but a paper was published about a month ago suggesting that 9-month-olds who go on to develop autism or respond with less to their name than those who don't go on to develop autism. So this was a very consistent finding of 12-month-olds, and there's been a recent demonstration that even as early as 9 months that this behavior may differentiate individuals who go on to develop autism from those who don't. And that's the earliest overt behavioral marker. I also bolded these at the bottom. This is work that I've done, that I did as a graduate student, identifying these 2 features that differentiate 12-month-olds who go on to develop autism. I'm not going to talk about that today. But I'm going to talk a little bit about some of the eye tracking work that we've done. And this work started 10 years ago in the context of this big study. We called it Infant Brain Imaging Study Network. Essentially, I arrived at University of North Carolina at Chapel Hill in the developmental psychology PhD program to study infant cognition and hopefully get in with the folks studying autism. And year after I arrived, this study started, and there was a postdoc working with this guy right here, John, who was working on eye tracking methods. And his wife got a great job at CHOP at the Children's Hospital of Philadelphia, and he left, and so there was this need for someone to come in and learn eye tracking methods. And so I kind of just slid in and developed an eye tracking battery for this study, which has really kind of launched my career. So it's a big group. Behavioral data collection has been ongoing for the past 10 years at University of North Carolina Chapel Hill, WashU in St. Louis, the Children's Hospital of Philadelphia, which is the Department of Pediatrics at UPenn, and University of Washington, Seattle. And there are also a number of support sites, University of Alberta, no longer the University of Utah, this is now NYU, the Data Coordinating Center at McGill, and then myself and a colleague, Jason, at the University of Minnesota. So what were some of the motivating data, the background data that motivated this study, the questions that would be asked in this study? So one motivating source of information, this was the first paper on infant SIVs published in 2005. Essentially, Lonnie Zwegenbaum and his colleagues at University of Alberta and colleagues across Canada developed what they called the Autism Observation Scale for infants. So essentially, they took what we know about autism in 2-year-olds and 3-year-olds and thought, if we were to try to capture some of that phenomena, those phenomena in 6-month-olds or 12-month-olds, what would we be measuring? So essentially, it's a standardized clinical assessment with the 6-month-old as they sit on mom's lap across the table from a researcher or a clinician, and you're trying to elicit certain types of behavior. You're trying to elicit social smiling, you're trying to play a peekaboo, looking for anticipatory gaze to the face, you're looking at how well they move their attention around space, a number of different items. So what they found was that there were no differences in infants who go on to develop autism at 6 months, but these differences emerged at 12 months, and they were sustained at 18 months. So this pointed to something very interesting happening between 6 and 12 months of age. back to that. So a couple of other sources of information that were important background for the study. In 2005, there was a report using retrospective head circumference data, so taking head circumference measurements and using that as a proxy of brain development, total brain volume development, to examine brain growth across the first 3 years of life. Essentially, the finding was that trajectories began to diverge right around the 12th month, such that infants who want to develop autism showed a slightly increased head circumference, slightly bigger brain after 12 months of age. And this, we didn't have this kind of timing, but we did have data from 18-month-olds, 18 to 24-month-olds, who came in and slept in the scanner, and we captured images of their brain and characterized total brain volume. And so this study actually characterized a stable rate of change of total brain volume from age 2 to 4 in young children with autism as compared to controls. But they were about 10 to 15% bigger. So this idea of brain overgrowth being a potentially early biomarker. What this study also characterized was the fact that brain volume, and I have a picture of this later, but essentially brain volume is made up of two things. It's made up of surface area and thickness. Okay? So you can identify each of those subcomponents, and each of those subcomponents of brain development is controlled by different genetic factors. And what they identified was that it's primarily surface area, increases in surface area that mimics this total brain volume. It's not cortical thickness contributing this total brain volume difference between groups. So we'll get into that in just a second. So the study design, we were enrolling infant siblings of children with autism. So we first verify that the proband, or the older sibling, actually has autism using our kind of gold standard research criteria. And then we see the infant sibling at 6 months, 12 months, 24 months, and then 36 months. And we do brain imaging and comprehensive behavioral assessments at each of these ages. And as I mentioned, so when I joined, I essentially had an opportunity to build in something experimental, something a little bit risky. My mentor at the time said, look, we don't want to trailblaze. The sample enough will be unique. Brain imaging is unique. Let's use a workhorse attention task. And so I told him that I was using something that was kind of a standard in the field, but really I innovated a little bit. And so the hypothesis, essentially, thinking about what's happening between 6 and 12 months of age. Well, a lot, for those of you who have spent any time with babies. So one researcher, Michael Tomasello, has called it the social revolution that occurs between 6 and 12 months of age. I mean, it's just fascinating how much is changing. So you get phonemic specialization. So infants who can discriminate between sounds, phonemes, and non-native language at 6 months can no longer discriminate sounds or phonemes in their non-native language at 10 months. So they're specializing with the languages that they hear in the house, right? So you get phonemic or linguistic specialization. You get specialization with face processing. 6-month-olds can discriminate faces in rhesus macaques, so and non-human primates very well, and in fact much better than adults, and much better than 12-month-olds. This changes between 6 and 10 months of age, okay, where you begin to get specialization, experience-dependent specialization with face. And then follow-up research has showed that if you actually continue to do book reading with lots of rhesus macaque faces, that you can extend this period of age. Fascinating. But there's more. There's more. So the onset of joint attention, which Michael Tomasello has also argued is kind of the foundational phenomenon that makes us human. Joint attention. It's a very simple behavior. It's when a caregiver will get the attention of the infant and redirect the attention of the infant to an object outside the infant's immediate visual field. That's the beginning, but that soon progresses into the infant actually redirecting the attention of the caregiver. And the idea is that the infant is acknowledging the agency and intentionality of the adult, acknowledging that the adult has a mind or a brain separate from the infant. So this is very exciting, right? So this is also happening in the latter half of the first year of life. A paper came out 3 years ago suggesting, and it's arguable, but it was published in Science, and so it got a lot of press, arguing that they had identified a neural correlate of consciousness, perceptual consciousness in 10-month-olds that they couldn't identify in 6-month-olds. So a lot is happening between in the latter half of the first year of life. The hypothesis that was driving the development of this task that I'm going to show you is, and the way that I was thinking about autism, is that essentially, in order to enter this period of specialization with social information, what's the prerequisite? What needs to be in place? And so I was thinking that, Michael Tomasello, who's done a lot of work on social cognition, he said, well, there's just this inborn, innate social motivation for the infant to kind of to prepare the infant for all these changes that occur in the latter half of the first year of life. I didn't know how to study social motivation in a 6-month-old, but I didn't know how to study attention. And I thought to myself, well, and with my grad school colleagues and my mentors, I thought, well, what if an infant needs to be flexible and efficient in how they move their attention in space as they enter into this period? And those infants who are less flexible and less efficient are not going to specialize with social information as typically developing infants do. So I developed a little task in which it's very simple. Moms bring their baby in and they're sitting in front of a computer display, and it's got some fancy eye tracking equipment that's going to record where the infant moves his or her eyes in response to what I show. And the display is very simple. Essentially, a central image appears in the display, and it's displayed for a variable duration of time so that we reduce kind of predictions or anticipations from the baby. And then one of two things happens. Either a peripheral target appears concurrent with the central, and we call this the overlap condition. And the target essentially overlaps with the central. Or in the gap condition, this central stimulus disappears and there's a brief period, 250 milliseconds, where it's a blank screen before this pops up. And essentially the idea is that these two conditions require processing in different neural circuits. So processing the gap condition is primarily going to be driven by oculomotor, or eye movement circuits, whereas you're going to engage some attention mechanisms in the overlap condition. So this is what the task looks like when we overlay gaze. The blue dot is gaze of a baby looking at this. It's pretty boring, but I got really good at getting as many trials as I could from babies. So essentially, all we're looking at is the time at which to initiate an eye movement to the periphery, and that's our dependent measure. So what did we find? So the first thing that we want to do is to identify whether individual differences in reaction time in this task are meaningful. So one way to do that was to test a model of whether individual differences in reaction time correlated specifically with individual differences in the part of the brain that I hypothesized were important for this behavior. So the gap condition, we had an idea about a specific white matter fiber bundle in the brain that should be more important for this task, and sure enough, the open circles represent latencies in the gap condition. So this is a sample of 40 7-month-olds who had brain imaging and eye tracking data. The open circles represent the gap latencies. The closed circles represent overlap latencies. You see there's no association between overlap and this fiber bundle, but there is an association here. So you know where we're going to the next question, right? So we go to the overlap condition, and we have hypotheses about a separate fiber bundle that's called the splenium of the corpus callosum. The corpus callosum is the biggest white matter fiber bundle in your brain. It is medially right in the center and connects the two hemispheres. And the splenium is essentially the most posterior sector, so it kind of courses out to the occipital cortex and to the parietal cortex, and to vision areas and to attention areas. So I highlight in yellow is what's called the gene with corpus callosum. We also measure that as a control track. And what you see is, again, we have an association between overlap latencies in this fiber bundle, but no association between gap latencies in this fiber bundle. So we think that we've identified something meaningful. We kind of have not a lesion-based double association, but essentially brain behavior double association that we're interested in. So essentially what we think we've done at this point is establish kind of the brain behavior phenomenon. Okay? The next question was, do patterns of visual orienting, or what we call oculomotor efficiency. And remember, these are going to be visual orienting is linked to the overlap condition, and oculomotor efficiency is linked to the gap condition. Do patterns of visual orienting or oculomotor efficiency differ between low-risk 7-month-olds? These are infants who have a typically developing older sibling. High-risk negative, high-risk infants who have an older sibling with autism but do not go on to develop autism themselves. And high-risk infants who do go on to develop autism. And what we find is we call this a disorder-specific effect, where the low-risk infants and the high-risk negative infants are both shifting attention or orienting attention faster than the infants who go on to develop autism. In about 40 milliseconds. It's not that big of an effect, but we think that it could have cascading effects on information processing. This is almost what we would call an endophenotypic pattern, but it's not, because this group in the middle, the high-risk kids in the middle, don't statistically differ from the low-risk kids or the high-risk positive kids. But the stair-step pattern, if we did get a significant difference, it would tell us that this oculomotor circuit, eye movement circuit, might be really, really meaningful. But because we got this disorder-specific effect in the visual orienting condition, the next question was, we have this in the behavior, we'd established the brain-behavior association in typically developing babies. So the next question is, does group moderate the association between brain and behavior? Okay. Are the associations, the brain behavior associations, different for different groups? And sure enough, they were. And so this is the simple slope that you've already seen, and this is the direction of the slope for infants who want to develop autism. And so this suggested to us that the functional coupling of brain and behavior that we observed in typically developing infants is not apparent in these infants who are going on to develop autism. And so essentially, this was one aspect of my dissertation that was published a few years ago. And so, a little interim summary on visual orienting. So, it's a domain general processing capacity that contributes to subsequent cognitive and social cognitive development. And we think at this particular time in development is especially important. And then one comment on the splenium of the corpus callosum. So this is a very interesting part of the brain. It's uniquely situated in the brain to contribute to experience-dependent cortical specialization. It's a lot of jargon. We think that it's a lot of information is flowing through the splenium early in development, and then it's kind of a gatekeeper for where information goes thereafter. It's also commonly implicated in visual perception and visual attention. So that'll get to our broader model in just a minute. So just to remind you, so this was, to get back here, so we think that some type of attentional inflexibility and efficiency in how infants move their attention in space may be particularly important right here at this 6-month time period. So what about total brain volume, total brain development? So we're going to look at total brain volume because this has been identified as a potential biomarker for quite some time. But we're also going to look at the independent components. So we blew up this picture of brain, and in between the white matter and the edge of cortex, we call this cortical thickness. And if we follow the blue line, this is how we're characterizing surface area. Okay. So we did this in about 400 kids, high-risk infants, but we had data from 6 months, 12 months, and 24 months in about 150 kids, about 15 who went on to develop autism. And we showed this, described this very interesting pattern of brain development. And this paper was published in Nature earlier this year. It looks a little messy, but that's because we wanted to include all of the data and show people what the data looked like. But essentially what we found is that at 6 months of age, there's no difference in these brain tissue structures. But what we observed was an increased rate of surface area growth from 6 to 12 months of age in infants who want to develop autism. This increase in surface area, this expansion of surface area, then predicted total brain volume growth from 12 to 24 months. such that by 24 months, infants who go on to develop autism have slightly bigger brains than infants who don't. And this rate of change in total brain volume growth predicted the social psychology at 24 months of age. So essentially, we felt like we described the brain overgrowth phenomenon in autism, and we're identifying some specific mechanisms for follow-up. Now, the regions of surface area expansion that were changing the most rapidly, interestingly enough, are in these areas where the splenium projects. So in the middle occipital and precuneus, these visual areas. So we think that this, we may be coordinating on some specific brain regions and specific behaviors that may be especially important for early identification. So just as kind of a summary model, at 6 to 12 months of age, we're seeing some of these sensory motor and visual orienting type deficits. We also see this hyperexpansion of surface area. But then by 12 to 24 months of age, we're beginning to see emerging autistic social behavior and this other phenomenon that's been fluororeplicated brain overgrowth. Okay, so now to the I have one slide on some of the most exciting work, I think. So what about prediction? So in this Nature paper, it's great to describe a phenomenon. We characterize the brain growth. But what else can we do? Can we use the brain information at 6 and 12 months to actually predict whether infants go on to develop autism or not? And so essentially what we did is, again, collaborated with our friends in computer science and implemented an artificial intelligence algorithm called deep learning. That essentially, we did a number of different configurations of this, but for this purpose, one of the ways that you can do this is you can split your data in half, and you can say, here are infants who go on to develop autism, and here's what their brain looks like at 6 months and 12 months. Here are infants who don't go on to develop autism. This is what their brain looks like at 6 months and 12 months. Now, the key to this is using a lot of data. So essentially, we use 78 surface area measurements and 78 cortical thickness measurements at 6 months and 12 months. So it's a lot of information that we're putting into this algorithm. And what the algorithm is going to do is it's going to iterate and find the best pattern of brain structure associations that differentiate the groups. So you do that, and you train this algorithm to do it, and then you apply the algorithm to a new sample to see if it actually works. And what we were able to do is identify with about 88% sensitivity and 95% specificity which infants would go on to develop autism at 24 months based on their tissue structure at 6 months and 12 months. And when we went back into the algorithm, what we found is that essentially it's surface area measurements that were the most meaningful in the classification. So what might prevention look like? So this was a motivating paper for me, and I think about this paper all the time. So this was written by Tom Insel, the former director of the NIMH, who was talking about trying to challenge researchers to think about strategic prevention. But in order to prevent, we need to discover what we think might be the causal pathophysiology, or what's causing this behavioral disorder, and this can be done at a great level. But it also requires revisiting assumptions about core features of psychiatric disorder. The vast majority of work is done on individuals who are already manifesting atypical behavior, right, or abnormal behavior. We know from studies of neurodegenerative disorders, such as Huntington's, that the brain changes precede the behavioral manifestation by sometimes 10 years. By the time an individual manifests the behavioral symptoms of Huntington's, 60 or 70% of the dopaminergic neurons in the ventral striatum have died. And so here we're borrowing from the neurodegenerative disorder literature and suggesting, well, let's identify brain features and brain differences that anticipate the emergence of behavioral disorders. So, so it also requires identifying a biomarker or behavioral marker that's sufficient prognostic sensitivity and specificity. And we think that we've done that. And then also the cost of prevention must outweigh it. So what might it look like? So we know that early intensive intervention works. And when I say intensive, I mean it. This is 4 hours a day, 5 days a week for 2 years. In 18 to 30-month-olds. It increased developmental quotient, which is the developmental equivalent of IQ, by about 15 points in the intervention group. It also, the same group, the intervention normalized electrophysiological patterns or activity in response to this, in response to familiar faces. So it normalized brain activity. And it also resulted in diminished symptoms as late as 6 years of age. This is really promising, but this is very intensive, right? So this is when we go back to the precision medicine model and think about dose. At what dose can we still make a difference? Does it need to be 20 hours a week for 2 years? These are important questions that we're going to ask. Now, recently, 2 papers have emerged where groups are taking infants at high risk for developing autism and doing an intervention with them. So they're not selected on any additional information. We just know that 1 in 5 or 1 in 7 or 8 of these children are going to develop autism. They're hoping to demonstrate kind of proof of principle that an intervention, preventive intervention perhaps, could be implemented as early as 8 months of age. So future directions. Well, we're scientists, so we need to replicate this, the pre-symptomatic prediction. We also want to be able to predict associated outcomes or more variable outcomes. We want to be able to predict language impairment, for example. If we can do that, then this is a specific target for an intervention. And then one future direction that I'm really kind of excited about learning about, because I know nothing about it, is we need to determine the ethical, legal, and social implications of pre-symptomatic prediction. So what are we doing on the Twin Cities campus? I got a grant to do some brain imaging in typically developing infants, and we're using cutting-edge hardware and software and acquisition sequences, and we're collecting the highest quality brain imaging data that's ever been collected. This is just a fun picture where we're mapping for the first time the thalamocortical connectivity in a 6-month-old. And hope with the idea of leveraging new technology and new innovation that will augment future studies of infants at risk for developing autism. We've got a great group. I wanted to highlight, if you guys are interested, and you write this down or I'll tell you later, we're having our UMAI Day. It's the University of Minnesota Autism Initiative on Friday, May 5th from 1 p.m. to 4:30. People are welcome to join us. You can go to this web address and register. We've got a great group of people working on autism at the University of Minnesota, represented by all of these different units. And essentially, it sounds a little funny, but I have active collaborations with people from each of these units. And we're really excited about the future of autism research in the state of Minnesota. So I want to acknowledge my lab, the eLab, and then the IBIS study. The IBIS network I started as a graduate student 10 years ago, and we just got a good score on a grant that we think we're going to be able to continue for another 5 years and follow our cohort of 400 infant siblings of children with autism into school age. So it's really an exceptional and unprecedented